Hello and welcome to today's GeneWiz Week webinar, Methods of Population-Specific Transcriptional Analysis of Brain Tissue. I am Damian Urena, Business Marketing Leader of Next Generation Sequencing Services at GeneWiz, and I'm very pleased to introduce today's presenter, Hope Cronman. Hope is a fourth-year MD-PhD student in neuroscience at the Icahn School of Medicine at Mount Sinai. She works in the laboratory of Dr. Eric Nestler, where she has helped refine sorting protocols for neurons and apply them in new ways to study the mechanics of the brain's reward system. Before entering into the MD-PhD program, Hope completed her undergraduate at Yale University, where she studied scientific and philosophical fields that make up cognitive science. If you have any questions for Hope, please enter them into the Q&A section, and we will address as many as possible at the end of the session. Without further ado, please welcome Hope Cronman.
Thank you, Damien, for that introduction. As he mentioned, my name is Hope Cronman, and I am an MD-PhD student in the laboratory of Eric Nestler at the Icahn School of Medicine at Mount Sinai, where I study the biology of stress and depression. The brain is a very heterogeneous organ. It is heterogeneous on many levels. The most obvious is indicated in the diagram at the bottom of this slide, which shows the distinction of all of the brain regions. These each serve different functions and are made up of different cell types. Furthermore, there is heterogeneity of circuits that communicate within and between these regions, and of cells within any given region. What's the relevance of this heterogeneity? Well, first of all, we are masking the effects of individual cell populations when we study whole tissue. If we have two cell types that have an opposite response to a given stimulus, we won't be able to resolve these individual responses. What we'll see is a net neutral when we look at whole tissue. Furthermore, psychiatric disease itself is very heterogeneous. Take depression, for example. Symptoms descriptive of depression are increased appetite or decreased appetite, increased sleep or decreased sleep, and so on. So no individual case of depression or other psychiatric disease is the same as the next. Therefore, it will be difficult to develop a blanket explanation across cell types for such a heterogeneous condition. And finally, the understanding and the resolution of this understanding will be important for the development of therapies as our technology for medication delivery advances. I want to talk about this heterogeneity in the context of the nucleus accumbens. The nucleus accumbens is a central reward integrator. That is, it receives reward-related information from numerous areas throughout the brain. It receives excitatory input from the prefrontal cortex, involved in executive control, the hippocampus, involved in memory and navigation, and the amygdala, involved in fear response. It also receives modulatory dopaminergic input from the ventral tegmental area in the hindbrain. So this picture is complicated enough as it is, but wait, it gets more complicated. If we look at the nucleus accumbens itself, we see another level of complexity. Again, as I mentioned, we see the heterogeneity of cell types within this given region. We see interneurons of various types, GABAergic and cholinergic, and importantly, on the left of this diagram, we see the major output cell type of the nucleus accumbens, the medium spiny neuron, which comes in two flavors. The first expressing dopamine receptor type 1, and the second expressing dopamine receptor type 2. D1 medium spiny neurons respond to dopamine by initiating an excitatory cell signaling cascade, while D2 medium spiny neurons respond to dopamine by initiating an inhibitory cell signaling cascade. So while both of these cells respond to dopamine, are GABAergic, send information out of the nucleus accumbens that's relevant to reward behavior, they have opposite functions and opposite cell signaling pathways. So to average their phenotypes is actually to mask individual changes. So in order to understand stress and depression, we want to examine the mechanism of change in the reward system at the resolution of populations of cell types. To do this, we've utilized a technology called fluorescence-associated cell sorting. And in what follows, I'm going to walk you through the process, an outline of the process that we use to isolate populations of cells. Now, we've used two separate techniques, one that isolates whole neurons and the other that isolates only the nuclei of those neurons. In order to do either, we use transgenic mouse lines. To do whole cell sorting, we use transgenic mouse lines which express fluorophores in the cytoplasm of the given cell type. We use one line that expresses a red fluorophore in the cytoplasm of D1 medium spiny neurons, and another that expresses a green fluorophore in the cytoplasm of D2 medium spiny neurons. So we first harvest the brain, we dissociate fresh tissue using an enzymatic dissociation protocol involving papain, and then separation on a Percol gradient to remove debris. And finally, we sort it through the fax machine to obtain a purified suspension of only transgenically labeled neurons. To sort nuclei, we use a different line of transgenic animals, and that's because the isolation of nuclei will involve the dissipation of the cytoplasm and any fluorophore that resides in the cytoplasm. Therefore, we need a fluorophore that is membrane bound and will remain in the nuclear membrane. So we've utilized D1 and D2 TLG transgenic animals. TLG is a membrane bound form of GFP, which does insert itself into the nuclear membrane. However, in recent months, we've piloted a new line of animals which expresses a fluorophore attached to a subunit of the ribosome, which still maintains signal upon isolation of the nuclear fraction. I will show you in the next slide some comparative data of the TLG lines and the RPL10A lines to indicate the increased purity of suspension that we get with the RPL10A animals. But briefly, the protocol for nuclear isolation involves harvesting brain tissue and then freezing of that brain tissue. Then we thaw the tissue, separate the nuclear fraction in a sucrose gradient, and sort through a fax machine to obtain a purified suspension of transgenically labeled nuclei. As I mentioned before, we've piloted two different lines for nuclear sorting. On the top, we see sorting of the TLG animals. This is the membrane-bound GFP isoform that I mentioned. We can see in P2 the population of transgenically labeled cells, but we see that it's continuous with unlabeled cells. Granted, we don't see the P2 population in the wild-type litter mates of the transgenic animals we sort from, but the distinction is not as clear between these two populations as we'd like it to be. However, if you look at the bottom of this slide, you can see a much clearer distinction with the RPL10A animals, and in the histogram on the right, a clear bimodal pattern. To compare whole cell and nuclear sorting, we have to take several things into account. Each has its pros and its cons. Whole cell sorting is, of course, inclusive of all cell fractions. It includes the nucleus as well as dendritic mRNAs. And if you're interested in studying, for example, the postsynaptic density, whole cell sorting is clearly the protocol of choice. Furthermore, in the whole cell sorting protocol, we don't freeze the tissue, and the cells therefore undergo fewer freeze-thaw cycles. Nuclear sorting has its advantages as well. If you're studying, for example, transcription factors, you want the increased resolution that this specific cell fraction provides. And if you are studying large numbers of animals, the ability to standardize is increased when you can add the freezing step. It allows you to process more uniformly large batches of samples. The cons of each of these methods are also notable. Whole cell sorting is difficult to standardize because it has to be performed on fresh tissue, and therefore it's difficult to perform a uniform dissociation across large number of samples. And also there's evidence that enzymatic dissociation of cells can induce transcription itself. This can be mitigated by the addition of inhibitors, NMDA inhibitors, tetrodotoxin, which inhibits sodium channels, and so on. Nuclear sorting is potentially a limited window into transcription because it might indicate transcripts that are withheld from translation in the cytoplasm. And this is an important point to consider when analyzing nuclear data. So we performed RNA sequencing on isolated populations of D1 and D2 medium spiny neurons, which were isolated using either whole cell or nuclear dissociation. And what we're looking at on this slide is a plot of the average read count per gene across the genome. On the left, we have nuclear sorting, and on the right, we have whole cell sorting. In each, red represents D2 and blue represents D1. While there isn't significant overlap in the lists of differentially expressed genes in the whole cell and the nuclear sorting conditions, we do see a similar pattern of transcription across the two protocols. That is, we see that D2 cells have fewer transcripts that are simply off, represented at 1 on the x-axis, and more transcripts that are lowly expressed, represented on the left side of the x-axis. However, comparing between nuclear and whole cell sorting, we can see that whole cell sorting captures more lowly expressed transcripts. To drive this point home, I'll call your attention briefly to the label on the x-axis. I have plotted here 0.5 to the average RPKM in order to capture transcripts which have a read count of 0, which we couldn't do with the standard notation of log RPKM. Therefore, anything from 0.5 to 0 indicates a gene with a read count, average read count less than 1. Those are genes that are expressed very lowly. And we can see, as I noted, that D2 cells in both nuclear and whole cell sorting have more of these lowly expressed transcripts. But you can also see that whole cell sorting in general captures many more lowly expressed transcripts. And we are heartened, at least, to see that the two methods showed similar transcriptional patterns of these cell types. So what can we do with sorted transcriptional data? There are several analyses that we can make. First of all, we can look at cell type enriched transcriptional networks and the regulators of those networks. And we can look at shifts in cell type identity. And I'll show you a few examples of these analyses. We analyzed transcriptional networks in D1 medium spiny neurons of stressed mice. And we plugged in transcriptional signatures to Ingenuity Pathway Analysis, which is a supervised machine learning program that uses protein-protein interaction databases to determine upstream regulators of the indicated gene changes. And what we find here is a specific regulator, a muscarinic cholinergic receptor, CHRM1, which is not indicated in D2 cells of stressed mice. This gives us a very resolved target for gene alteration in stress. Furthermore, we can look at changes in cell type identity. The analysis that I've performed here is called rank-rank hypergeometric overlap. Simply, it analyzes the similarity in ranked gene lists between two sequencing lists. On the x-axis of both of these plots, we have D2 versus D1 medium spiny neurons at baseline. And on the y-axis, we have stressed versus control neurons, either D1 or D2 type on the left and right, respectively. And the key in the upper right hand of this slide indicates the method for analysis of these diagrams. Heat in the bottom left quadrant indicates genes that are upregulated in both conditions. Heat in the top right of the diagram indicates genes that are downregulated in both conditions. And in the other two quadrants, we have the alternative comparisons between gene sets. So what we can conclude from this is that D1 cells in the condition of stress are downregulating genes that are important to D1 MSN identity at baseline. To home in on that analysis a little bit, look at the leftmost diagram here, and we see heat in the top right corner. That means that genes that are down in the D2 versus D1 baseline comparison are also down in the D1 stress versus D1 control comparison. So we can see that D1 medium spiny neurons in the condition of stress are losing their D1 identity, whereas, shift your attention to the right now, we don't see any heat in the D2 comparison, from which we can conclude that the changes happening to D2 MSNs in stress, of which there are many, are not changes that are relevant to D2 identity determining transcripts. And this conclusion is borne out by the Venn diagrams below, which show the overlap of up and down regulated genes in the compared conditions. On the left, we see that the totality of genes downregulated in D1 medium spiny neurons during stress are contained within genes that are important for D1 identity at baseline. On the right, we see minimal overlap of genes changed in D2 medium spiny neurons after stress with those that were important to their identity at baseline. So these analyses are pointing us towards cell type specific targets for manipulation in stress. But what about the next level of resolution? What about single cell resolution? So now we'll be moving from tens of thousands of cells to single cells. There are several platforms on which to make these analyses. There's the C1 platform from Fluidigm with its new HT chip can analyze up to 800 individual cells. The Chromium platform from 10x Genomics, which can analyze in one run up to 10,000 cells. And it's the commercialized version of DropSeq from the McCarroll lab at the Harvard Medical Center. All of these platforms utilize microfluidic design to isolate single cells, and they have their own pros and cons, which I would be happy to discuss in the Q&A. And the analysis of single cell data can yield some very interesting results. Typically, people utilize the t-distributed stochastic neighbor expression analysis to determine the distribution of populations of cells in a sample of thousands of single cells. And this analysis can reduce multidimensional data, that is 10,000 individual cells with completely different transcriptional profiles, to something that looks like this, something that's two-dimensional and allows us to visualize the separation of cell populations. And while this looks straightforward, there are many remaining questions with these analyses. First of all, how many clusters should we be discerning? Are we sure that the three green clusters seen on the left of this diagram are in fact representing different populations of cells? And when you get into the nitty-gritty of this analysis, you can see that you are able to manipulate the parameters and determine how many clusters the algorithm will output. So it becomes a relevant question for the bioinformatician to ask, what resolution of cell populations do we want? Furthermore, it's been notoriously difficult to determine doublets, that is, individual samples that capture two or more cells. The way that this has been done in the past is to look at individual cells and see if they express known markers of two or more different cell types. But as you can imagine, this is somewhat circular logic. It doesn't necessarily lead to the discovery of new cell types. And finally, there's room for improvement in the comparison between samples of distribution of cell types. All of this being said, the question is an open one. What level of resolution of cell populations is relevant for therapeutic change? We believe that the functional differences in populations as distinct as D1 and D2 medium spiny neurons will certainly be important for understanding and regulating the function of a network within a region like the nucleus accumbens, but we don't yet know what level of single cell resolution will actually add to our ability to dissect these circuits. So this is the next frontier, and we're excited to explore it. And with that, I would like to thank all of my lab members, especially my mentor, Eric Nestler, and of course the funding we receive as a lab and as an MD PhD program at Mount Sinai. Thank you so much for listening. I'm happy to answer questions, and I would love to communicate further about the specific protocols I've used for cellular dissociation and analysis of sorted transcriptional data.
Thank you, Hope, for a very interesting presentation on methods of population-specific transcriptional analysis of brain tissue. We'll now take a few questions from the audience. Our first question is, what controls do you use to assess that your transcriptional patterns are not impacted by your cell sorting technique?
That's a great question. Thank you for asking. So it's shown in the literature that different methods of sorting do activate cellular transcription. To different degrees. In fact, a recent paper from Fred Gage's lab showed that whole cell sorting, more than other techniques like nuclear sorting or trap-seq, activate immediate early genes in the cells being sorted. So yes, it's definitely an open question. We are confident based on those analyses, the protocol we're using does activate transcription to a lesser degree. But of course, we want to make sure that all of our samples undergo the same sorting method. So we have internal controls within the experiment to compare the treated and untreated samples, each of which undergo the same dissociation protocol. So we would hope to see the same transcriptional effects of sorting in both the control and experimental populations.
Thank you, Hope. Our next question is, how do these FACS-based techniques compare to trap RNA-seq?
Another very good question. So I didn't mention trap at all in this presentation, but our lab does currently have an ongoing project comparing FACS-based methods to trap methods. Now, like nuclear data, the trap method has been shown to have a lesser degree of transcriptional activation than whole cell sorting. So in that sense, it's more like the nuclear data. But I think, and this goes back to what I was saying earlier in the presentation about pros and cons of sorting methods, it really depends on the question you're asking. So trap, of course, purifies RNA from ribosomes. So that means RNA that's actively being translated. So I think that a technique like trap would be most relevant to a question about active transcription and maybe a more acute stimulus. So again, Damien, I think this goes back to the question that you're asking experimentally.
Thanks again, Hope. We have a third question here. Depression is estimated to occur at a higher frequency in females overall. Do the expression patterns for your cell types show differences between females and male mice when compared?
Great question. So as we all know in the scientific world, NIH funding depends increasingly upon the analysis of sex differences for any question that we're asking. And this, like every other question, nearly every other question in the brain, does exhibit sexual dimorphism. That is, D1 cells in males do look different than D1 cells in females. However, we have not yet characterized these differences, and that is upcoming. I would say broadly, yes, we expect differences. What those specific differences are, we don't yet know.
And we have time for one last question. Can the cell sorting protocol be used for all types of cells in the brain?
Yes, in fact, it can. So I mentioned earlier in the presentation that our lab uses specific transgenic lines of mice. That is for the whole cell sorting lines, which have under a specific promoter a cytoplasmic fluorophore, and for the nuclear sorting lines that have under a specific promoter a fluorophore which embeds in the nuclear membrane. So any of these tools can be repurposed as long as one knows a cell-specific marker. So if you have a cell marker, you can generate a transgenic line and follow the same protocol. And we have optimized the cellular dissociation for isolation of neurons. So it's possible that certain parameters would need to be varied. And I'd be happy to talk with any individuals who are more interested in glial sorting, for example, than neuronal sorting. And I see very quickly that there's one last question, whether this technique can be used for other model organisms. I think that's a great question, and I don't see any reason why it couldn't be. I just imagine that you would have to pool more samples. So from one mouse, we get 2 punches, each a millimeter thick, 14 gauge in diameter. And from those 2 punches, we can isolate approximately 20 to 30,000 nuclei or whole cells. And that is more than sufficient for the RNA sequencing. We found we can perform RNA sequencing on as little as a 5,000 cell sample. So depending on the recovery, the rate of recovery of neurons from these organisms, I think that you would just have to modify the pooling element of the protocol.
Thanks, Hope. This is all the time we have for today. I would like to thank Hope Kronman again for a great webinar. Along with our webinar series, we also have a lab startup program, GeneWiz credit awards, and exclusive promotions available this week. Please check out genewizweek.com for more information. Thanks again, Hope Kronman, for a great presentation.